So it is 2. We will get started. The first thing that I want to show you is how to access your test if you're having trouble doing that on Blackboard. So let me do that. Just make sure I'm on the right page. Okay, so let me share my screen here. All right. So here we are looking at our Blackboard page, and you can see that student preview mode is on. What that means is I'm looking at this page like a student would. So what you need to do is you go over to the left where it says My Grades. When you go to My Grades, you go all the way to Test 1. Once on test 1, you should see your calculated grade here. Here, I haven't graded my own test, the one I just submitted as a student, so that's why you saw the exclamation point. But if you click on your score, you should be able to see your answers, so your selected answer is here, and the correct answer. So just about every question should have the correct answer underneath it. Some don't, like when lysing a cell to purify a protein, what 3 challenges are there and how do you overcome them? So those, I was grading those based on what people put, since there's could be a lot of things that you put, but everything should have the correct answer. So that is how you get to the your test to look it over. Now let me pull this up. Okay, PowerPoint. So for test 1, the average was 65, so not the greatest in the world. Standard deviation, 24, which means that scores were all over the place. What you see in this graph here is just the distribution of letter grades in our class. So you can see this is basically what you call a bimodal distribution. Those of you who have taken stats, you basically have a curve right here and a curve right here, which isn't that all uncommon, because if we look at fall of 2019, the average for our test 1 was 65. So I got the same exact average last year as I got this year. Standard deviation, more or less the same. 23 versus 23.9. Distribution of grades, basically the same. The only difference between last year and this year, the percentage of people who got A's and B's seems to be flipped, but otherwise it is more or less the same. All right, so that's just to give you some stats perspective. Those of you who did not do so well as you thought, I will be nice, and whatever is your lowest test score, I will drop it. When I calculate the grades, I will just drop the lowest test and calculate your grades like that, and it's all automatic, so I'll do that for you. So what to do if you got a score that was not where you wanted it to be? Well, so you saw how my tests are laid out, hopefully. And when you go back over the test, what I want you to realize is that, the majority of the questions that I take for the test, I give you the vast majority, in that we go over most of them in class. So most of the questions on the test we went over in class as those questions. So when I'm asking these questions in class, what I'm really doing is I'm showing you what I'm going to ask you on the test, right? So that's why when I pause and do those pauses to let people think about it. I really do want you to be working on it during that time, right? This is me giving class time to you all to try and just do those problems, get ready for the test. I took some tests from questions from the reading guide as well, just looked at those, and I think I just copied and pasted like one or two. And then from the old test, that buffer problem, where you have lysine, that protein, half protein, that is straight ripped from that test. All I did was take, instead of D, I put K. And that question was actually asked in class on Friday, much to my amazement when that was asked. Not on Friday, but on Wednesday. So if you were paying attention to lecture on Wednesday, you should have saw me solve that question. And I gave you the answer right there, pH equals pKa. But yeah, all the answers are there. And let me just say also, biochemistry is a level 4,000 class. It's supposed to be hard. There's supposed to be a lot of information, right? A ton of information in a short amount of time. And keep in mind, this whole semester, we're only covering like not even half the textbook, right? So don't, try not to beat yourself up. Try not to be too far down if you do not do so well. Just see how you can improve, right? Look at your study habits and ask yourself, did I study all throughout the weeks, or did I wait till the last couple of days to try and pack in all this information? Because look, I was a student too, right? I know how it goes. You come to class, you write down notes, you don't really pay attention all that much. Then the last 3 days you try to study, right? For this class, that's probably not going to work because there's just too much information. So just keep that in mind, and really just aim. If you were somebody who didn't do as well as you thought you would, no, aim at that improvement, right? You've seen test 1, you see how I asked the questions. Expect more of the same on test 2. When I ask questions in class, you can expect to see that on the test. Well, like I said, biochem's hard. It's not supposed to be easy. If it was easy, everybody would be a biochemist. It's taking 2 subjects which are hard on themselves, chemistry and biology, and combining them together to make a hard subject, right? This is a difficult class. And you might not want to hear this, but honestly, for every single class we do, you should be spending 2 to 3 hours outside of class studying. That's what it takes to learn this stuff. So every single week, you should be looking to put in a minimum of 6 hours on your own studying. And like I said, I know I was a student. I didn't do that, but that's kind of what's expected. If you ask any science teacher, any of your teachers here at Texas A&M San Antonio, they will give you the same answer. How long should I study? 2 to 3 hours outside of class for every class that you do. So just keeping that in mind. This class is not that great for cramming. Any questions in general? Did you take off partial credit if wrong information was included in the essay questions? For the essays, I have some of those for scores, right? Where I'd say like what I'm looking for. If the information was like wrong, I didn't give you negative points, I guess, but I didn't. So the one example I can think off the top of my head is why do water and oil not mix? Hint, just saying the hydrophobic effect is not sufficient. So if you put like water and oil repel each other because they're different charges, that's wrong. That's not how it works at all. And so I would not give you full credit for that. So yeah, there was partial credit given, I guess. I thought we didn't have to go into much detail with some questions, just the main idea. I mean, so the question I can think about that is the ELISA question. So for ELISA, if you just said antibodies, they bind, that does not tell me how ELISA works. So I guess that's the super main idea. But if I ask like explain how something works, that's me looking for details on, do you know how this works? Yeah. So the question, I'm afraid to include everything I know. So I'm not going to mark you off for putting too much right stuff. If you put just extra stuff, I'm not going to like ding you unless that stuff is obviously wrong. So just, yeah, that's what I would say. If you want to say more and you think it's right, say it. Because a lot of the times you might back your way into a right answer. Just be careful, because if you just spew out everything and what you say isn't correct, you're not going to get full credit. Yeah. I was thinking more of the DDNTPs. Oh yeah, so for that question, I was looking for two things. One, did you know it stopped the reaction? Two, did you know that they're labeled? Because if you just said that stops the reaction, well, okay, but because I think the question was like, what is the purpose of DDNTPs in this method? And it's twofold. One is stops the reaction, two, they're tagged. So if you just said they stop the reaction, yeah, that's part of it. So you got partial credit, but they're the signal that tells you what the amino or what the nucleotide is. So I was looking for two parts on that. If you don't put the extra stuff, you can't get the extra stuff wrong. Yeah, or you might not be completely right. Okay, so let's put that behind us. I am amazed it is the exact same average as fall, though. That just shows you that like doing this over the computer really doesn't have an overall effect on you as a population. It might have effect on you as the individual, but as a population, it seems to be the same. Gotta know your amino acids. That's all I can say about skeletal structures. All right, but moving on. If you remember way back on Wednesday, before you met my first test, we were talking about how to purify proteins. And we talked about a bunch of different columns. Were we still going to go over the most missed questions still? So there's actually, I thought there was, I don't have a good way to know where people got everything wrong. So what I would say is I have the answers up there. So you can go back, and if you don't understand why something's wrong, let me know. I'll be free to just send me an email or something, or if you want, we can have a in-person chat, and then we can talk about, why is that? Why was that wrong? What's the correct answer? So I'm gonna go about it like that, just because we're already behind, and there is not an easy way for me to do that unless I go back through all the tests, and I graded these over the course of like 4 or 5 hours. So I took breaks, so it's not all in my mind when I was doing it. All right. So anyways, we're purifying proteins, right? And we're using different columns. We talked about ion exchange columns. We talked about gel filtration columns. Now the next column we're going to talk about is affinity chromatography. And an affinity chromatography column is like the column you want to use if possible. And what affinity chromatography is that your beads have some kind of ligand or ligand, however you want to pronounce it, has some kind of ligand in it. And your protein of interest will bind this ligand very tightly. And why this is great is because ideally, only your protein binds this ligand and nothing else does. As we see in this example, where I have 3 different proteins, red, blue, and green. Red's the one we want. Red's the only one that binds the ligand. Blue and green comes out, right? So all of these interactions are non-covalent. And so this is actually something I want to bring up in the test, right? So if you see the word intermolecular, that means non-covalent. So intermolecular interactions are not covalent bonds. So non-covalent interactions, so you don't form any covalent bonds through this. It's all electrostatics. It's all hydrophobics. So it's just that non-covalent interaction. And what you do is that you change your solvent, your elution conditions, to make your protein not bind the ligand anymore. There's 2 main types that are really useful. One is immunoaffinity chromatography, and that's where you get an antibody on the bead, and your protein will bind to this antibody. Similar idea to ELISA in that antibodies are extremely specific for one binding site, so only your protein should bind to the antibody. A much more common type of affinity chromatography is called metal chelate. In this, your beads are either zinc or nickel. You have metal as your beads. And proteins that have metal chelating groups, such as multiple histidines in a row at the end of the terminus called a his tag, what will happen if this is my metal, the histidines will just go and non-covalently bind this metal. So usually for a his tag, you put 6 histidines in a row, and hopefully we all know what histidine looks like. And if you know what histidine looks like, you can kind of imagine how it would bind. But you put like 6 histidines in a row at the N or C terminus, your protein sticks to that. Anything that doesn't have these histidines will go right through. So just a little review question here. How do I get 6 histidines onto a target protein for this to work? How do I ensure my protein just has like 6 histidines on it? What can I do using biochemistry to make sure that happens? By the way, if you're still not sure what histidine looks like, right? And so you have these like ring-like structures, and they just interact with the metal. Add metal. So what I want is my protein, right? And I want either a bunch of histidines at the front or histidines in the back or histidines in the front. The question is, how do I ensure that? What did we talk about last week, maybe 2 weeks ago now, that will allow us to do such things? Remember, histidine is just an amino acid. See, isn't it great now that we learned our amino acids? I can just put his, and you should know what I'm talking about. Learning amino acids is fun. Slice the puppy in there. Do you want to use more technical terms? I guess splice the puppy in there is, I know what you mean, I think. But what tools would we use? So you have your DNA, right? And histidine is just DNA. Yeah, use vectors, use PCR, right? And what you want to do is just, use PCR on your vector that has your protein in it and encode 6 histidines, encode either to the N terminus or the C terminus, right? That's how you add anything to your protein. You have to modify the DNA. So don't forget the whole idea of vectors and modifying DNA and all that. But any questions about affinity chromatography? Not, we will move on to our next subject. Yeah, so I have a DNA sequence as my protein. How do I add more amino acids to it? Well, it should be in a vector. Then you use PCR to insert the histidine sequence to your vector. Yeah, you just use PCR because PCR not only replicates DNA, we use it to mutate DNA. So if there's not a perfect fit, you can force things in there. So it might take some trial and error to get that, yeah, you can use PCR to put in any sequence you want into DNA. That's one of PCR's main uses. It's not just to replicate DNA, it's to change DNA. And then to produce it, you need a vector, put it in E. coli, all that good stuff. Yes, use a, so usually you wouldn't use a vector to insert histidine, right? The vector, let me get my pen back out. The vector, right? Hey, no, please. Pen, please. Thank you. Your vector will already have your protein sequence in it. Then you, with your PCR primers, you engineer your primer to put in a hist tag, like 6 histidines in a row. So what you can do, if you know your protein starts here, right, you can make your primer span that region, and in the middle, just add a bunch of hist tags, and it should work. I mean, you need your protein sequence in DNA form, and you need a way to express it. So that's going to be in a vector. I guess technically you don't. If you want to modify an organism's genome to add a hist tag, that's a lot harder. It's possible using CRISPR-Cas9, but that's well, how do you grow a protein in E. coli, right? You need a vector with your protein sequence, right? That's what we talked about like last week or a little bit before that. So we have a vector, we have our protein DNA sequence. How do we modify that? Well, to modify the DNA of a vector, you use PCR, right? And with PCR, you can mutate amino acids because if it's not a complete fit, so what I mean by complete fit, right? So this is my DNA. If my primer fits perfectly here and here, but not perfectly here, your primer will still bind. And then when you replicate the DNA, the polymerase will add these new nucleotides. And when you add these new nucleotides, you make a new amino acid. Yeah, the vector has the protein of interest that we amplify with PCR. The primer tags in the PCR process will add the histidine tags, yes, because it's not a perfect fix. So that's how you create mutant proteins. Yes. Vector has the protein of choice, and then we'll PCR to insert the change by having our vectors. That's the great thing about PCR. This is just not for duplication. It's also for changing stuff. Yes, that's like one of the great things about PCR. Biochemists use that all the time. You want to know how an amino acid works in a protein? Mutate it to an alanine. See what happens. Does your protein not work anymore? Then that amino acid was super important for it to work. That's a typical like biochemistry experiment. That's like one of the first things you do. Okay. Now we need to talk about SDS Page. SDS stands for sodium dodecyl sulfate. You don't need to know that, but that's what it stands for. SDS is also soap. It's a detergent. And what you do with SDS Page is that you use this technique to separate proteins by mass, where heavy proteins go at the top, small proteins go down the gel. The basic of this is that SDS is negatively charged. And your protein, if this is your protein, it just gets filled with this detergent molecule. And what will happen is that it will unfold and be coated with this negative charge. Then with this gel apparatus, we send electrical current down the gel. And so your proteins start up here, and then they'll migrate through the gel because they're all charged, they're all negatively charged. And the further they migrate based on their size. The smaller they are, the further they migrate, because they're going through a gel, right? So gels have holes. The smaller you are, the easier it is to go through holes. You also need to add 2-mercaptoethanol, shown down here. You don't need to know the structure of that. What 2-mercaptoethanol does is that it breaks cysteine-cysteine disulfide bonds, which we had a question on the test. So cysteine, if you remember, has SH for its side chain. Disulfide bond is 2 sulfurs being added, or forming a covalent bond. So just so we have this down, for my 4 proteins, rank them from least farthest movement to most farthest. So if I give you 4 proteins on an SDS page, tell me how they will migrate. SDS is not safety data sheets, no. Sodium dodecyl sulfate. Sodium, 12 carbons for dodecyl, sulfate for SO4. One, 4, 3, 2, 1, 4, 3, 2, 2, 3, 4, 1. Yeah, so the least farthest, so maybe it was how I asked the question, but the least farthest is protein 2, right? So that will go the least farthest. Then 3, there we go. So it should go, protein 2 will be at the very top, then protein 3, then protein 4, then protein 1. So if I ask this question on the test, I will have to be a little more specific on least farthest to most farthest. But yeah, if you're big, you can't go through holes that easily, so you get stuck at the top. If you're small, you can go through holes super fast, and you go on the bottom. And what we see is that on an SDS page follows this nice logarithmic movement, where how far you move is based on the log of your size. Again, you don't need to know that. That's just how it works, though. Any questions about that idea? Okay. There is also something called native page where you don't add SDS. But yeah, that's a similar idea, just a little bit different. What we're going to talk about, though, is 2D gels, 2G gel electrophoresis. This is where you're separating by two things. Now I can see the protein on the bottom got cut off, so I'm going to rewrite that for you in a second. So in 2D gel electrophoresis, you have a gel. And this gel is just a giant pH gradient going from left to right. So pH 1 so the gel is like pH 1 down this strip, pH 14 like down this strip, pH 7 down the middle, and then 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, all the way across, right? And so what you first do is what's called isoelectric focusing. And what that means is that you're going to apply a charge going from left to right, right? And you're going to see where your protein stops at. Now, in an electrical field, when are you not affected by an electrical field? If you're a protein, when do you think you stop being affected by an electrical field and you stop moving? When you are neutral, when your pH is the same as your pI. pKa is a similar idea, but not quite. We're talking about pI here, isoelectric point. pKa is the acid constant, but it's a similar idea though. So yeah, your pI is the pH where you are neutral. You don't have an overall charge. So if I apply an electric field, you're going to move until you reach the pH that is equal to your pI, and overall you have no charge. If you're not charged, you're not going to move in an electric field. Then you do SDS Page and you sort by weight. Again, low weight at the bottom, high at the top. The idea, why would you ever use this? The idea is, let's say that you have a cell type that's never been studied and you just bust it open and you just want to see all the proteins in there. So you just separate all the proteins from DNA and lipids and all that stuff. You do this 2D gel electrophoresis, and each point is an individual protein. You can then cut these proteins out of the gel, send them for sequencing, and now you know every single protein inside that cell. That's one application for it. But let's see if we have this. So let me rewrite this. Protein 5 got cut off. That has a weight of 32 kilodaltons, isoelectric point of 8. I want you to draw a square, label the left corner pH 1, then pH 14 at the right corner, and tell me where you're going to find these 5 proteins on your square. So using 2D gel electrophoresis, I will give you the answer to this in a minute or two. But see if you can figure that out. All right, let's try to make sense of this. Let's just start with protein 1. All right? Protein 1 has a pi of 5.3. So it'll be stopped roughly at a, well, not roughly, it will stop at a pH of 5.3, which is, I don't know, like right here, maybe. And it's 32 kilodaltons. So it's a small protein, so it will travel far down the gel. So it's roughly there. Protein 2 has a pi of 8, so it'll be farther to the right. It's super heavy, so it's going to be at the top. So that's 2. Protein 3 has a pi of 13.2, so it's even farther to the right, but it's lighter than protein 2, so it's going to be a little farther down, so we can say 3 is right there. Protein 4 has the same pi as protein 1, so it has to be in the same vertical row, but it's heavier, so it's going to be farther up. There's protein 4 right there, and it has to be below 3. I just made my square too tiny. Protein 5 has the same weight as protein 1, the same pi as protein 2, so I have to line that up, and protein 5 is going to be roughly there. So that's how this 2D gel electrophoresis works. You travel to where your pi equals to your pH, and then you're separated by weight. What is a maximum KD? On the gel, each gel that you can order different gels from just suppliers, and they will tell you what your maximum and minimum weights are for separating, linearly. You have a linear separation. Different gels have different weight ranges, and that has to do with the percentage of acrylamide in it. So the more acrylamide you have, you're not going to be able to separate big things well, but you'll be able to separate small things very well. The less acrylamide you have, you'll be able to separate big things well, but not be able to separate small things. So I think, like, this is just going on a little bit of tangent, but I think what you shoot for is like 12.5% acrylamide, bisacrylamide, if I'm remembering right. And this will give you like a good enough range to separate most proteins. If you need different ones, you can always make or order your gels differently. So what one do we follow, PI or KD? Both. PI is going from left to right, KD is going up to down, right? So your PI, you find by going left to right. Your KD is up to bottom, where large KDs are at the top of the gel, small KDs are in the bottom of the gel, because that's SDS Page, this idea. And we're just adding isoelectric focusing, where we have a electrical charge where whatever the protein's PI is, you stop at that pH. PI along the x-axis, weight along the y-axis. Yeah. Mass determines how far it will travel. Yes. The heavier you are, the less you are traveling. Because the heavier, the way to kind of think about this is, imagine that like there's a bunch of ropes and they're just randomly like across from each other, right? And you're a snake. And let's say you're a tiny baby garden snake. You're able to get through these holes a lot easier than, let's say, a 6-foot python. That python is going to get tangled all up in these holes, in these nets, and it's not going to be able to get its way through very fast. While baby garden snake can go down the thing super fast. So small proteins go down further in the gel than big proteins. How does a gel achieve a pH gradient from left to right? I've never made one of these, so I don't know technically how it's done, like inside the lab. But you can order gels that have been prepared where different section of the gels have different pHs. I'm guessing it's somehow done with how they pour it and how they make it. They make it under different pHs. So I never been privileged to actually, make one of these. So I don't know technically how it's done. So unfortunately, I can't answer that question too well. Do know it does exist though. So that is a good question. Unfortunately, I do not know the answer to that. But it's in the gel. I believe it's in the gel itself. All right, so that's the last one for this PowerPoint. Let's move on to the other PowerPoint. Of course, we're not going to get through it all day, all of it today. That's fine. There's a section on here that I can skip that will save us some time. So no worries about that. So I want to go to this. And that, yeah, I didn't change the date. It should say Monday the 21st. But I didn't change it from when we moved everything back a day. Okay. So we're going to talk about how to analyze proteins now, because we just talked about doing that 2D gel electrophoresis activity. And like I said, when I was talking about 2D gel electrophoresis, one of the great things about doing that is that, like, from a tissue or a cell, you can get every single protein in that cell. The hard part is, what are these proteins? That's what we're going to talk about now. So the first type of experiment you can do is called the Danzel chloride method. And this is an N-terminus analysis. You figure out what your N-terminus amino acid is. So let's take a look at this. So here's my Danzel chloride. This is my reagent. And I add that to my polypeptide. Some vocab for you: anytime you see polypeptide, that basically means protein. They mean the same thing, more or less. And what you do is that you add acid. Well, first, you just add a base and it will attach to your polypeptide. I'm getting a little ahead of myself. So you attach it to your polypeptide in a basic condition. Then, with acid and high temperatures, you break your peptide and you'll get a danzelated amino acid. Danzelated amino acid. Now, what's cool about a danzelated amino acid is that they are fluorescents, and you can determine what amino acid it is based on what wavelength of light you get out. So each amino acid will have a slightly different wavelength of light, right? And so whatever amino acid comes out of this N-terminus analysis, whatever fluorescence rather, you can determine what amino acid it is. Is there differences between a polypeptide and a subunit? Yes. A polypeptide is a protein. A subunit is part of a protein. Yeah, that's one of the ones that you can get into the weeds a little bit, because all subunits are polypeptides, but not all polypeptides are subunits. Yes, subunits are like just a part of a protein, while polypeptide is a much more general term for amino acids connected by a peptide bond. That's what a polypeptide is. Subunit has like function to it, has structure. Polypeptide doesn't have to have structure, doesn't have to have function. Only the terminal will be danzelated. Yeah, for the most part. There is one amino acid that could also be danzelated from this. Can anyone figure out, now that you know all 20 amino acids memorized, right? What amino acid may also accidentally be danzelated? Not isoleucine, not proline. What amino acid that's in a peptide bond Alanine. Now you're just guessing. Alanine. What determines if you can be dancylated? That's a good question. An NH3 group, or I guess an NH2 group. Gluon or asparagine? No. Lysine. There we got it. Lysine. Lysine, if you remember, ends in an NH3 plus. To be dancylated, yes, special K. You need, I hate biochemistry books always doing this, but you need an NH3 there. I guess when you add base, it will become an NH2, but that's kind of what you're looking for. So lysine can also accidentally be dancylated on its side chain. So, but usually you control for that. You know that that could happen. See, learning amino acids is fun. Comes up so often. Now I can just say lyse and it should be like, yay, NH3 group. All right. But other than finding the first amino acid in a peptide chain using this method, why would we ever do the dancyl chloride method? Because I have other methods that I can determine the first polypeptide of easily. Why would I use this specifically though? What could be the main use for the dancyl chloride method? Can anyone think of that? It's not obvious. This is one of those big brain questions. So if I have a test that's good at finding N-termini, and I have a new protein that I've never seen before, I might use this method on that protein. There's other methods that are more popular now, but I could use this method. So this method does not tell you how much glycine there is. I'm not sure why you think that. This method is only good for N-termini. It does not work for glycine. I said lysine. Lie. But it won't tell you how much lysine's in there either. So you can add a different amino acid to the end later. No, usually you just use this for sequencing. When you do this, you blow up the protein. It's not usable anymore. It's only for sequencing. Knockout to see what happens. If you want to do a knockout, you want to knock out DNA, not a protein. So this method, like I said, is only good for sequencing. And freeze the other amino acids. No, again, once you break all these peptide bonds, that protein's not usable. So this method is good for finding one amino acid only. To blow up proteins, no, that's super easy to do if you wanted to. We as humans have gotten quite good at blowing up proteins through other means, like weapons. So we don't need to really do that. Okay, so this is a good little thing. Every single protein or every single peptide starts with an N-terminus. It has to. Every peptide. Proteins can be made of more than one peptide. Proteins could be made out of like 2 peptides coming together, or 3, or 4, or like 26. That's not unheard of, where you just have like 26 different peptides forming together to form like the super protein. I've seen things up into the 40s. Probably more than that if I would think about it. This will tell you how many N-termini you have. So one of the things you can do when you discover a new protein is figure out how many N-termini there are, because that will tell you, that's one of the ways you can determine how many polypeptide chains there are. How many different amino acid chains are coming together to form one protein. Now, honestly, nowadays you would just probably use SDS Page to do this, because SDS Page would also tell you this. But this is another method that could be used to determine how many peptides there are. You can have more than one. Yeah, proteins, we'll go over this when we talk about quaternary structure. But yeah, proteins, a lot of proteins are made out of more than one polypeptide chain. There's only one N-terminus per polypeptide chain. So polypeptide chain is N-terminus all the way to C-terminus, and it's everything that is in a peptide bond. You can have multiples of these, though, coming together and forming one protein. That happens all the time in nature. We'll talk more about that when we talk about our proteins, though. Yeah, the answer is determine how many N-termini are in a protein. That's like the other reason. I mean, reason number one why you'd use this is to sequence the N-termini. Reason number two, to find out how many polypeptide chains are in your protein. Not really used that I know of all that much. That doesn't mean people don't do that anymore. You can use SDS Page, but this is a way, it's always better to do 2 experiments to confirm one thing. So like if you do this plus SDS Page, and you find out in both of my experiments, I have 3 different peptide chains making one protein, that's pretty strong evidence. It's hard to dispute that. While if you just did SDS Page and said, I have 3 different proteins, people could always come back and argue that. Like, well, how do you really know you have 3 peptide chains? Do another experiment. And then you do another experiment, you see I have 3 peptide chains, and it's like, there you go. Thank you, reviewer. Thank you for wasting 2 months of my life. Not that that's ever happened to me or anything. Not that I have strong feelings about reviewers for papers. Anyways, that's enough of me ranting. That is the end of our discussion today. Like I said, what I'm probably going to do on Wednesday is skip mass spec. I'll talk about what it is and how it works, but I'm going to stop. I'm going to skip the questions because the questions take like 15 minutes. So we'll get caught up via that, just skipping mass spec. Otherwise, so the amount of N-termini to figure out how many polypeptides. So for every polypeptide, there's 1 N-termini, right? So if, like I said, I got, this is going to be hard. I have 2 polypeptides. They come together to make one protein. If I discover this protein, I can't tell there's 2 polypeptides unless I do some kind of experiment. And so if I cut off each little tip of my finger, and I have 2 tips of my finger, that means, oh, 2 fingers made this protein. So that's what it can be used for. Polypeptide chains are different than polypeptides. No, polypeptide chains and polypeptides are the same. I just think of these, I guess I used the word chains and not even thought about it. Yeah, so basically I think of these like kind of like limp spaghetti, and it's way easier to say chains in that case than polypeptide limp spaghettis. So they mean the same thing. So if you ever hear me say polypeptide chain, which I will do a lot, just not thinking about it, it's polypeptide. It's the same thing. Any more questions? Yes, end of the noodle is the N-termini. Well, you have an N-termini and a C-termini, right? So this pen is my peptide. The point, oh, let's do that. The point is the N-terminus. The back end is the C-terminus. All my amino acids, except in reality, peptides are never this stiff. They're wet spaghetti. They are moving all over the place. Then they fold up into a beautiful 3D structure. But we'll talk about that shortly when we talk into proteins. My favorite subject of this whole semester, talking about protein and protein structure, because that is what I did for my PhD work. That in biophysics, but we don't have a biophysics class. Anyways, I'm rambling and I have nothing important to say other than I'll put a homework assignment up. Again, if you didn't do so well in the test, it's in the past. crying about the past does not change the future. So just learn from your mistakes. Biophysics for biology topics spring 2021. That would be fun. I think you would not like me so much because biophysics is super hard, but it would be fun. All right. But anyways, I will see you all later. If you have questions, as always, feel free to email me. Yeah, take care.